Question 2 is to do a metabotropic neurotransmitter receptor, and we want it to be inhibitory. Presynaptic cell would be the same. I won't draw the details again because I just drew them all up here. And we'll release neurotransmitter, this time dots. Let me zoom in again. And here is our postsynaptic cell. In this case, the receptor will not be an ion channel. Most commonly in this class, we're going to talk about G-protein coupled receptors. They actually typically have 3 subunits. I usually just draw 2. They activate a G-protein, a subunit of the G-protein comes off. And adenylate cyclase is one of the common targets. Adenylate cyclase converts ATP to cyclic AMP. So here is AC. AC I usually draw tethered to the membrane because commonly adenylate cyclase is a membrane protein tethered on the inside of the membrane. Then our cyclic AMP will bind to, for example, a calcium channel. I'm sorry. Our cyclic AMP could bind to a calcium channel if we wanted more amplification, but cyclic AMP might bind to a chloride channel or a potassium channel, whatever there is. And binding to that channel then would open up, for example, a chloride channel. Chloride comes in, makes the cell more negative, that spreads all the way to the axon hillock, and some of that negative charge would reach in. Again, as the charge spreads, there's a cluster of negative charge in here, less and less gets further and further away, until at the axon hillock, we may just have a little bit of actually reaching the axon hillock. But with multiple synapses and with multiple action potentials, that could build up. And of course, the synapse nearer to the axon hillock will have a bigger effect on a potential action potential. So ionotropic, the receptor is an ion channel. Metabotropic, the receptor is not an ion channel. G-protein coupled receptors are one common version, and the pathway goes through a second messenger, such as cyclic AMP, to open a channel, and finally we bring in this case, negative charge. We could have made it a potassium channel, in which case potassium would have left. The next question was to sketch some neurons showing divergence, in which a single neuron controls multiple targets. So this is commonly shown with a circle to represent the neuron. We could draw some dendrites out of it. A single neuron can have many dendrites with lots of branches, but commonly when we're showing neural circuits, we just know that it has dendrites. And if we want to show some other neuron having an effect on it, we just send an axon branch to it. So here is neuron A. And we want divergence, in which a single neuron controls multiple targets. So we can draw the single neuron to start with, and then multiple targets, B, C, and D, all doing something different. So we draw an axon branch to B. A neuron only has one axon, so you don't want to do multiple axons coming out. That would be wrong. But you can have as many branches on the axons as you want. So here's divergence. If we send an action potential down this neuron, it'll go down that branch. It'll also go down this branch, and in fact, every branch to release neurotransmitter at every single one of these. So we have one neuron A that could cause action potentials in B, C, and D. Next is facilitation, in which it requires at least 2 presynaptic neurons to cause action potential in a single postsynaptic neuron. So here we've got a single postsynaptic neuron, and we need at least 2 presynaptic neurons. So I'll just draw all 3, and then I have to figure out how to connect them. So we're going to want an action potential in the postsynaptic neuron. So here's where we're going to have an action potential. We want both of these to synapse on C. So how are we going to get requirement, how are we going to show a requirement that both of these are required to get an action potential in C? Well, we know that it can take more than one episode of neurotransmitter release to get enough positive charge in to accumulate at the axon hillock to start an action potential. So let's imagine that this synapse from A gets enough positive charge in to get us plus 15 millivolts at the axon hillock. In other words, we were inside here at the axon hillock, we were minus 70 millivolts, but from A, we get plus 15 millivolts. So if we add those together, we're at minus 55 millivolts, not threshold yet. But if at the same time we had action potentials from neuron B, and some of that charge, again, dispersing through the whole cell, but some of it makes it to the axon hillock, and that also was plus 15 or even plus 10 millivolts, depolarizing for 15 millivolts. Add these two together, we're at minus 40 millivolts. That is threshold. So below minus 45, so we're going to get an action potential. And an easy way to show that on your neural circuits is just to show it here, plus 15, plus 15. And that will tell me that what you mean is that not here, but here at the axon hillock, it's plus 15 from each. If we were at minus 70, we'll get to minus 40. What if you wanted 3 of them? Well, if you needed 3, then instead of plus 15 each, you might do plus 10, plus 10. That would take us from minus 70 to minus 60, minus 50. And then if we had another one that was plus 10 or even plus 5, that would get us at the axon hillock to minus 45, and we'd get action potentials. So this is how you can show a need to combine, facilitate from multiple neurons to get an action potential. Okay, brain areas. Cortex, thalamus, hypothalamus, medulla, cerebellum, pons. And with 1 to 4 of the major functions of that region of the brain, I often put medulla and pons together. They're both foundational brainstem parts. And then we also want somatic nervous system and autonomic nervous system. Okay, so let's draw a brain and a spinal cord. If you want to figure out where the eyes are, here are the eyes. So cortex is this outer region. Should actually be bigger, but I'm not going to worry too much about all that. The thalamus, the hypothalamus, cerebellum, the pons, and medulla. And then the spinal cord down below, and going both out and in, we'd have the somatic nervous system, and autonomic nervous system. The autonomic nervous system, somatic nervous system is voluntary muscles, so all your skeletal muscles, all the movements you make that you're in control of. The autonomic nervous system is divided into the sympathetic, which is often called fight or flight, and it basically energizes you. It sends blood, for example, to brain, heart, and muscles, and does other things that get you to pay more attention, to have more energy for movement, to be prepared for sudden movement. That's all the sympathetic. The parasympathetic is more maintenance. It sends, for example, blood to liver, kidney, intestines, makes your somatic muscles more relaxed in general. Parasympathetic is for maintenance and support and repair. Sympathetic is for action, attention, focus. It's not necessarily fight or flight. It can just be excitement and delight, but you're paying more attention. And it does hormonal things and also sends blood to areas that get you more ready to move, to be more active. These brain areas, pons and medulla, a lot of basal functions like breathing, regulating heart rate. The hypothalamus regulates a little bit higher level things like body temperature, sexual maturity, and reproduction, hunger, aspects of sleep, and so on. Thalamus is a relay and pre-processing center. So it is passing messages from other areas out to the cortex and back, and it's doing some processing. For example, it takes visual information and does some pre-processing to do things like help you know which things in your vision are near each other in space, and then sends information back to the visual cortex to be processed for you to recognize images and color and shapes and movement. The cortex then does these higher functions. So cortex has some high-level processing and decision-making areas and planning. It has your major sensory areas, so motor control and touch, visual processing. Along the sides is auditory, hearing and speech processing, many memory functions, and so on. So what I want is for you to have a sense of how you get inputs from all over the body, from sensory neurons, both from sensory neurons on your skin, sensory neurons in your eyes, your hearing, but also internally from your organs, such as your intestine and your stomach, where you're aware of at least some of it. And then some of that is conscious and some is unconscious. So some of it you get as stimuli up here, some of it is just processed by the autonomic nervous system. And then you have inputs out to things that regulate hormones and regulate functions that you're not consciously aware of. You don't consciously control that sudden activation of fight or flight and the adrenaline release and the blood flow changes and so on. But your decision making up here and your motor control areas, I forgot to mention cerebellum is especially important in motor control, coordination and fine motor control and some aspects of motor memory. And these then go out to somatic nervous system to control your voluntary muscle movements.